What are cells and what do they do? My name is Rusty Owenbrink. I'm a board-certified osteopathic family physician with over a decade's experience as an integrative physician. And we're going to talk about basic biology in this review, starting with what our cells are and what it is that they do and how they do it. Basic cell biology. There's some basic concepts such as cell theory. In 1838, Schleiden and Schwann proposed that the cell is the smallest form of life, if we consider viruses, prions, etc. as cells. Cells arise from pre-existing cells. All forms of life are made of cells or collections of cells. Since then, we've learned that certain cells are differentiated for different functions. Some cells secrete chemicals outside of themselves to control their immediate environment or perform specific tasks. Fibroblasts, for instance, make collagen fibers which strengthen tissues. White blood cells secrete cytokines which influence other nearby cells, recruiting more white blood cells to the site of the infection, etc. Cells are classified as fundamental units of structure and by how they obtain their energy. Prokaryotes are the typical bacterial cells. They have no nucleus and contain a cell wall. Eukaryotes, that's us, those are animal cells, and that's what we're going to study today. Energy intake is one way to classify different types of cells. Autotrophs are self-feeders. They use light or chemical energy. An example of this would be plants. Heterotrophs are other feeders. They obtain energy from autotrophs or from other heterotrophs. Again, that's us. Many bacteria and animals are heterotrophs. Multicellular organisms are created from a complex organization of cooperating cells. These cells require communication on a cell-to-cell level and also need to be regulated. Activities such as acquiring fuels, expelling waste, and communications within the organism have specially evolved systems that develop to coordinate the various functions of the cell. Size and scale is an important concept to grasp in this evaluation. You'll see from the diagram above that electron microscopes can see things smaller than light microscopes. Resolving power is an important concept to understand. The greater the resolving power, the smaller two dots can be and still be picked up as two separate dots. At less of a resolving power, they will look at one dot or even be invisible at all. Chemistry deals with structures that are invisible to the naked eye, vastly smaller than what biology deals with in terms of the ability for visual observation. The scale above is basically listed on an order of 1,000, so that a nanometer, nm, would be 1,000 angstroms, and a micron would be 1,000 nanometers, a millimeter would be 1,000 microns, a centimeter would be 10 millimeters. The smaller molecules will be on the order of size of 1 nanometer, or 10 to the minus 9th meters. A meter is about 39.36 inches long, a bit over a yard. A nanometer is 1 billionth the size of a meter, 10 to the minus 9th. A micrometer is 1 millionth the size, or about 1,000 times bigger than that. Viruses will range from 50 to 100 nanometers in size. Bacteria are about 1,000 times larger at about 1 micrometer, or 10 to the minus 6 meters. Animal cells tend to be about 10 micrometers in diameter. Plant cells will be about 100 micrometers in diameter. Light microscopy can resolve objects a bit under 1 micrometer apart. Scanning and transmission electron microscopy, however, can zoom down to about 2 nanometers. Methods of observation. Light microscopy is a standard way in which cells are evaluated for gross structural features. Before a cell can be examined, however, it needs to be fixed, killed and preserved, and stained with special chemicals that stain specific cellular components. Many cells would be transparent, thus invisible on microscopy. Differential staining allows certain structural components, types of protein, carbohydrate, and fat, to be stained specifically in different colors to enable easier examination. This is the concept of differential staining. The Wright's stain is commonly used to evaluate blood cells. Gram stains are used to evaluate bacteria, which are classified as gram-negative. They will appear red, or gram-positive, blue, which gives clues as to which antibiotics will work against them. Hemolysin and eosin are typically used to stain animal tissues. Other stains can also be used, including fluorescent stains, radio-labeled stains in which radioactive compounds are added to them to look for specific chemicals. Electron microscopy also uses various staining techniques of its own. These are based on heavy metals. And the electron microscopy is done in a vacuum setting, so there's no way that electron microscopy could be done on a living animal cell. Cellular components and structures are the next things that we'll talk about. Not every cell will have each of the structures described in this review, but we would give an idea of what a composite cell, a cell that contains all the structures, would look like. The diagram below is a composite to show typical cellular components. Muscle cells would have linear structures of actin and myosin. Neutrophils would have numerous lysosomes and vacuoles. Other differences between all different cell types exist. This is a stylized typical cell. There's nothing typical at all about it. This is a figment of our imagination. But all these different structures inside the cell are basically called organelles. And we're going to review what these organelles do and how they work, and give you a rough idea of what chemicals are involved in each of them. Biological membranes. Membranes are dynamic complexes of proteins, carbohydrates, and lipids. The membranes are fluid with components that can move, change, and perform vital physiologic roles. They allow cells to communicate with each other and with their environment. Membranes are important for regulating ion and molecular traffic flow between cells and into and out of individual cells. Defects in membranes and structure cause many significant. Biological membranes are composed of a phospholipid bilayer, as noted in the picture to the right. The hydrophilic ends are green. They face outward to the blue water in the extracellular and in the intracellular spaces. The hydrophobic ends are yellow. They intermix with each other. These hydrophobic ends repel water, but they react very well amongst themselves. There are also specialized areas within the membrane, such as receptors for extracellular agents, which can include hormones and cytokines, which are all different forms of cellular communication signals. Membranes are differentially permeable. They allow some things to pass, but others do not pass. There are numerous chemical receptors on the surface. These are too small to be seen by light microscopy. There are also ion pumps embedded into the membrane. These pumps burn energy by converting adenosine triphosphate, ATP, into adenosine diphosphate, ADP, plus an inorganic phosphate, PO4 group, which releases energy that's now available for metabolic work. The energy is used to power ion pumps that continuously pump sodium out of the cell while pumping potassium into the cell. This maintains a concentration and electrical gradient from inside and outside of the cell. These gradients are a form of stored energy. The stored energy can, under certain circumstances, allow an electrical impulse to travel down the length of the cell when certain pores open, allowing sodium to flow in, potassium to flow out, calcium to flow in. They cross into or out of the cell. This crossing of the ions travels as a wave down the length of the cell, called an action potential wave. Muscle cells can be quite long. Nerve cells even longer, up to over a meter in length. Once the impulse passes by, the ion pumps go back to work restoring those gradients that were disrupted as the wave of the action potential passed by. The interior of a cell membrane is approximately minus 60 millivolts compared to the outside of the cell, due to this concentration gradient and the action of these ion pumps. Cell membranes also contain specific compounds made of proteins, lipid, and carbohydrate that could be thought of as antigens that trigger our immune system to react against them or to recognize them as being part of our self. Some of these antigens also have other chemical roles. There are also receptors for a variety of polypeptide hormones. Sterol-based hormones, on the other hand, are lipid-soluble. They pass right through the cell membrane and go into the nuclear membrane to interact directly with the nucleus of the cell. Cellular structure we're going to refer to is known as the cytoskeleton, which gives the cell framework and structure. This is an example of the differential staining that we talked about with microscopy. In this sense, actin molecules are stained red. Green is the stain for the microtubules that make up the skeleton, and blue is the stain for the nucleus of the cells. Looking at this picture, you can see that there is definitely an internal framework within each of these cells. You can see the blue nuclei and the structures around them that give the cell its shape. These internal workings are unique to eukaryotic cells. They're not found in bacterial cells per se. They're highly dynamic. They allow significant changes in the appearance, dynamic behavior, and functions of the cell. The cell is a very dynamic three-dimensional structure filled with cytoplasm. Long fibers of the cytoskeleton are polymers made of smaller units that repeat. The primary cytoskeletal cell skeleton fibers are microfilaments. These are fine thread-like protein fibers that are 3 to 6 nanometers in diameter. They're composed of a contractile protein, actin, which is the most abundant cellular protein. Myosin is also associated with actin and interacts with the actin to allow cellular gliding, contraction, and cytokinesis, or cell movement. Actin and myosin are also responsible for the contraction of muscles. The actin and myosin is complex together in a linear arrangement in this case. Microtubules are another form of cellular structure. They're cylindrical tubes about 20 to 25 nanometers in diameter. They're composed of protein subunits called tubulin. These tubulin have alpha and beta subunits to them. They provide movement to cilia, flagella, and propel cells such as spermatozoa flagellum, or fluids such as cilia in the lungs, which beat the sputum up toward the mouth. These microtubules also act as a scaffold to determine the cellular shape, provide tracks for intracellular organelles such as the endoplasmic reticulum and the vesicles, which we'll discuss later, to move on. They allow for cytoplasmic streaming, such as what happens with white blood cells move through capillary walls to reach pathogens and germs. Vesicles are also involved in this movement, such as neurotransmitter-containing vesicles that are necessary for our nerve cells. Movement of pigmented vesicles also happens for camouflage. And these microtubules are also involved in cell division. The cytoskeleton also includes cellular motors. These can be either actin and myosin or dynein and kinesin, which are components of the microtubules, that interact and burn adenosine triphosphate to ADP and inorganic phosphate, causing motion. Duchenne's muscular dystrophy is an example in which cellular motors don't work properly. There are also cross-linking units and nucleation promoting factors. Microtubules also separate chromosomes during mitosis or cell division. When arranged in geometric patterns, there are 9 peripheral and 2 central microtubules inside the flagella and the cilia that provide locomotion. There are also intermediate filaments. These are about 10 nanometers in diameter, providing tensile strength to the cell. The nucleolus is the next portion of the cell that we will examine. It lives within the nucleus of the cell, where in this diagram, number 1 would be the nucleolus, number 2 is the nucleus. The function of the nucleolus, it forms around specific chromosome regions within the nucleus. It's composed of proteins and RNA, or ribonucleic acid. This is where transcription occurs, and it modifies the ribosomal RNA to help create ribosomes. It acts as a cellular metabolic stress sensor, regulating ribosomal RNA production based on the cellular environment. Nucleus contains the genetic material of the cell organized into chromosomes. In human, there are 23 pair of chromosomes, including 1 pair of sex chromosomes, either X or Y, that determine gender. Females will have 2 X chromosomes. Male will have an X and a Y. Genes are regions of the chromosomes that contain DNA code required to make specific proteins such as enzymes, structural proteins, etc. There are also transcription regulating portions of the DNA that don't necessarily code for a protein but help to determine when these proteins are transcribed and made. The nucleus is surrounded by a membranous nuclear envelope. The nuclear envelope has regulatory pores that allow messenger RNA or mRNA to leave the nucleus for protein formation and to allow entry of other compounds such as ATP, nucleic acids, proteins, regulatory factors, and fat-soluble vitamins and hormones to enter the nucleus. The nucleus has its own nucleoskeleton similar to the cytoskeleton of cells. Histones are proteins that help fold and package DNA into the chromosomes. Human DNA laid end to end would be about 2 miles long, yet it all fits into a very small space thanks to these histones that have a lot of positive charges, amine groups, that help to fold the DNA into place. What does the DNA convey? There are 4 base pairs: adenine, thymine, guanine, and cytosine. Think of these 4 as letters in an alphabet. Individual amino acids are defined by 3 base pairs. Think of a 3-letter word. If we were to print this out on paper and put the human genome, all of our genetic material, into a book, that book would be over 1 billion words long. It could be bound into 5,300-page volumes. The book would fit into the nucleus of a cell smaller than a pinpoint, and all 5,000 volumes are contained in every cell as a strand of DNA that's over 2 miles in length. Every cell has this. Telomeres are little bunches of DNA at the end of the chromosomes. These diminish over time in number throughout the life of the individual. There are those that think that these telomeres are part of what determines our lifespan, that they correlate with lifespan among many different species. Some species with short lifespans don't have many telomeres. Species with long lifespans have a lot more of them. The next structure that we will explore are called ribosomes. These are the primary sites of protein synthesis. A ribosome is composed of ribonucleic protein in 2 subunits. There's a 40S and a 60S subunit which combine to make an 80S unit. This size is smaller than the expected 100 due to some overlap. Each subunit is composed of ribosomal RNA, rRNA, and protein. Transcriptional RNA, tRNA, carries the appropriate amino acid to merge with the 70S subunit for each amino acid nucleotide triplet match for the encoded protein synthesis. So a base pair, the codon of 3 base pairs will come out. Each different 3 base pair triplet will specify which amino acid is to be used. That will be on the messenger RNA that the ribosome forms up on, and as it moves along the messenger RNA, it will select a 3-letter sequence, determine which amino acid is needed for that, and the appropriate transcription RNA will bring the amino acid into the ribosome to be strung onto the previous amino acid to form the primary structure of the peptide. The small subunit reads the messenger RNA sequence. The large unit then plugs the correct amino acid in the 3-base sequence designated by the messenger RNA. The peptide chain grows as the ribosome progresses along the messenger RNA. This is basically what it would look like. You can see the messenger RNA that looks like the little gear tooth things. The ribosome would be coming along. Then above that, you see the pink balls of the growing peptide chain. Ribosomes are structurally part of the rough endoplasmic reticulum, or RER. This keeps the structural components in place for easier protein synthesis. If these things were floating loosely in the cytosol, the milieu within the cell, would be very hard for things to happen and to be all in one location to line up where they're needed. Having them membrane bound makes it structurally easier to create proteins. The rough endoplasmic reticulum is a cellular organelle that is interconnected network of flattened membrane enclosed sacs and tubes known as cisternae. These membranes are part of the rough and smooth endoplasmic reticulum and are continuous with the outer membrane of the nuclear envelope. The outer cytosolic face of the endoplasmic reticulum is studded with ribosomes for protein synthesis. These proteins are released into the cellular milieu, or the cytosol. This is a diagram of what the rough endoplasmic reticulum would look like. You can see numerous folds within the cell, the endoplasmic reticulum, with little dots representing the ribosomes that are attached to the reticulum. The smooth reticulum, on the other hand, is very smooth without the ribosomes being attached. And you can see, if you look carefully, both types of reticulum, endoplasmic reticulum, are covered there. To the left of the nucleus, you see the ribosomes, and then in the lower right-hand corner of the cell, above the lysosome, you see smooth endoplasmic reticulum. Smooth endoplasmic reticulum lacks the ribosomes. It functions in lipid and carbohydrate metabolism and in detoxification. The next structure we'll review are the vesicles. Vesicles look very much like small cell membranes with an intracellular component on the outside and an intravesicular component within the inside. It's a small organelle consisting of fluid enclosed in a typical lipid bilayer membrane. These can also be called liposomes. These cells secrete by exocytosis by forming the contents of a given compound, for instance a neurotransmitter that's formed within it or stored within it, to be released into the extracellular space. It will travel by way of the microtubules to the cell membrane. In a nerve cell, that would be at the synapse that the microtubules would carry it to. And then when the action potential came along, the vesicle would fuse with the cell membrane, releasing the contents, typically a neurotransmitter, within the vesicle into the synaptic cleft. Liposomes can also be used for cellular uptake, phagocytosis and endocytosis of different compounds, as a white blood cell would do when it's eating the debris of a bacterial invasion, or for other use within the cell. Vesicles can carry out many functions. White blood cells engulf then break pathogens. This is a Golgi body. These package proteins inside the cell before they are sent to their destination for secretion. It's a form of a vesicle. This form of vesicle is specifically designed for exocytosis. We also have vacuoles, which are similar to vesicles but are found in plants, fungi, protozoa, and some animal and bacterial cells. This is essentially a larger vesicle generally formed by the fusion of several smaller vesicles. Lysosomes are other types of vesicles. Lysosomes come from the Greek root lysis, loosen, and soma, or body. These are spherical vesicles containing hydrolytic enzymes that can break down biomolecules, such as nucleic acids, carbohydrates, proteins, and lipids, as well as cellular debris that contain all of these components. These lysosomes will contain over 50 different enzymes, all active in an acidic environment maintained within the lysosome. It would be toxic to the cell if that acidic environment were within the cellular milieu of the cytosol. These are responsible for homeostasis and secretion, plasma membrane repair, cell signaling, energy metabolism. The centrosome is an organelle that is a microtubular organizing center. It's composed of microtubules organized to separate chromosomes during mitosis and meiosis. And if you look closely, you'll see that there is two separate clusters of microtubules oriented perpendicularly, anchoring each other. and from there the microtubules branch out. The cytosol is the intracellular fluid cytoplasmic matrix. This is the liquid inside of the cells that contains all the organelles. It's bound within the cell membrane. It contains all the organelles. This is where parts of cellular metabolism take place. Some of it happens in the cytosol, others in membranous organelles within the cytosol. This is a complex mixture of water and salt ions, including potassium, magnesium, sodium, and chloride, and others. 67% of the water within our bodies is contained in the intracellular cytoplasm. The cytosol environment must be carefully maintained in terms of water, osmolality, pH, constituents in the milieu, etc. With dehydration, water leaves the vascular spaces of the body. Then the interstitial area between the cells, finally it comes out to the intracellular water. 45 to 60% of our weight is water. An approximate distribution would be intracellular, 35%, interstitial fluid, the fluid between our cells, are about 12%, and the vascular fluid, or the plasma within our blood vessels, would be 4 to 5%. With loss of water, osmolality increases, altering the carefully balanced orchestra of life that keeps us going. Clearly, water is very important. pH is also very important. The skeleton acts as a long-term buffer, donating alkaline minerals such as calcium and acidic minerals such as phosphorus to help maintain that perfect balance. The lungs provide the most rapid pH correcting by breathing fast to blow off carbon dioxide to make us more alkaline. For instance, if we're becoming too acidic by creating too much lactic acid with anaerobic or stressed out metabolism, which will be covered further. The kidneys provide intermediate term balance of osmolality and pH by regulating the flow of ions into the urine versus keeping them in our bodies. Dietary proteins contain acidic sulfate, phosphate, and other groups. The kidneys are critical to maintaining many of our balances. The mitochondrion is the last of the organelles that we will discuss. This is what it looks like. It's a fascinating structure. These are thought of as the power plants within the cell. It's a membrane bound, not a cell membrane, but a mitochondrial bound structure that is actually pretty autonomous. It reproduces all by itself. It comes from the mother. So when you were born, your father, or when you were conceived, your father contributed sperm, your mother contributed an egg, and the egg contained mitochondria that are copies of her mitochondria. There are different pores in different places, the cristae, the matrix, and a very complex membrane system within. And this membrane system holds numerous enzyme systems that help to metabolize and create energy by burning fuel and creating ATP and NADH. The mitochondrion is the cellular power station where energy is converted and power is generated in the form of ATP. This is the organelle that generates the power by converting fuels, such as fats, into the ATP, which is the energy currency of the cell. Size of the mitochondrion is from 0.5 to 1 micrometer in size. It contains numerous inner membranes which harbor the membrane-bound enzyme systems responsible for power generation. As with cellular membranes, there is an electrochemical gradient between the inner membrane of the mitochondria, the cristae, and the outer membrane of the mitochondrion. This gradient is part of the power-producing process of the mitochondrion. The inner membrane encloses the matrix, which itself contains the ability to self-reproduce. It's got its own DNA there. The mitochondrial DNA is similar in some respects to bacterial DNA. There is some speculation that evolutionarily larger multicellular organisms engulfed and parasitized or developed symbiotic relationships with energy-forming bacteria that later adapted to intracellular life as mitochondria. The mitochondria contain independent ribosomes for protein synthesis, and those inner membranes form folds known as cristae. During reproduction, as I mentioned earlier, the mother's mitochondria are passed to her offspring. The ovum contains the mitochondria which self-reproduce. The number of mitochondria in a cell is variable depending on how much energy that particular cell needs. For instance, athletes will have a lot in their muscle cells, heart muscles, etc. The mitochondrial energy pathway is where we're going to be heading next, and this is where it gets a bit complicated. There is aerobic respiration, which uses oxygen, and there's anaerobic respiration, which does not use oxygen. Anaerobic respiration can be thought of as being much less efficient. You don't get as much power out of anaerobic respiration as you do with using oxygen in aerobic respiration. Again, this all occurs within the mitochondria. This is the overall summary of mitochondrial metabolism. And don't worry, folks, we're not going to have a test on this, but it does give you some idea of how complex it is. There's all these different enzyme systems in different stages and phases where the ATP is generated from the ADP and NADH is generated from NAD. There are a variety of different cofactors that are involved in this. The electron transport chain is mentioned in the lower left-hand corner. You can see references in the upper left-hand corner to glucose metabolism, degradation of fatty acids, metabolism of amino acids, and fatty acid synthesis at the top. Glycolysis is the breakdown of glucose. It's also known as fermentation. This is an anaerobic process. It does not require oxygen. So the way this works, glucose is phosphorylated, which takes up energy. It's energy negative to the cell. The cell spends energy to do this. It's converted to fructose 6-phosphate and then to fructose 6,1,6 biphosphate. Again, it uses more energy to get another phosphate going. From this point forward, however, the energy is restored back to the cell. The net energy yield of glycolysis is 2 ATPs generated from ADP. That basically gives us back what we invested into the cycle, and also 2 NADHs, which can then further be oxidized to produce more ATP. It will also, the glycolysis process will also yield either pyruvic acid, 2 of those, or acetic acid, or alcohol in some cases, depending on which enzymes are involved and which organism is doing it. This breaks down carbohydrate, but it will not break down proteins and lipids. It utilizes the services of many vitamins. Vitamin B1, thiamine, as thiamine phosphorylase, releases the carbon dioxide. Vitamin B2, riboflavin, combines with the FADH2 to help convey electrons into the tricarboxylic acid cycle, or the Krebs cycle, which we'll go over in a few moments. And then from there, it moves them into the electron transport chain, which we will also review in the future. Vitamin B3, niacin, is nicotinamide adenine dinucleotide, NADH, transports electrons to the second stage of glycolysis. Vitamin B6, pyridoxine, is where stored energy is broken down into accessible energy forms. 2 molecules of ATP are produced to convert glucose to 2 molecules of pyruvic acid or pyruvate, producing 2 ATPs, which were used to get the process going anyway, and reducing 2 NADs to NADHs, which are then shunted to the oxidative phosphorylation electron transport chain to produce ATP. Fermentation, as I mentioned, can also produce lactic acid and ethanol. Glycolysis is a pretty inefficient way to get energy. It produces at about 2% thermodynamic efficiency. ATP contains 7.3 kilocalories, and the waste products, either pyruvic acid, lactic acid, or ethanol, still contain energy. They need to be broken all the way down to carbon dioxide, which requires oxygen to get that energy into the tricarboxylic or Krebs cycle. During periods of intense physical exertion, however, our muscles will go anaerobic. They will form pyruvic acid, which will then be converted in the liver back into glucose and glycogen, which is stored in muscle and liver for future emergency anaerobic use when our metabolic demands are exceeded by what we have to supply. This is the Krebs cycle, the tricarboxylic acid cycle. Again, this is a rather complex cycle, memorized by college students and med students the world over, but we're not going to worry about memorizing it. Just take a look at it. You'll understand that there are several different places where energy is metabolized, oxygen is used, a variety of intermediate stages during the cycle. This whole cycle will happen bound to membranes within the mitochondria. And again, it uses oxygen and produces water and carbon dioxide as net products with ATP for energy use. It can also metabolize lipids and proteins. And it utilizes the services of the following vitamins. Vitamin B1 thiamine to decarboxylate pyruvate and convert to alpha-ketoglutarate. It's also a transketolase reaction in the pentose phosphate jump. Vitamin B2 is FAD, which is reduced to FADH2. It's used to transport electrons. B3 transports electrons in the NAD cycle. The N is B3. B5 involve all reactions that use coenzyme A that's frequently used with the lipid metabolism. B6 converts amino acids and glycogen into suitable fuel. B7, pyruvate carboxylase, brings pyruvate into the tricyclic acid cycle. B9 is a free radical scavenger to prevent mitochondrial damage. B12 converts odd-chain fatty acids into succinate and homocysteine into methionine for free radical scavenging and mitochondrial production as a methyl or a CH3 donor. Overall, this cycle produces 1 ATP, 3 NADHs, which are then used to form more ATP, 2 carbon dioxides. It has about a 30% efficiency versus the 2% for glycolysis. Next, we'll talk about the mitochondrial oxidative phosphorylation or electron transport system. And this is, again, a rather complicated slide. There's more information available at the website if you're so inclined. But basically, the citric acid cycle is producing a lot of NADH that will then be oxidized to NAD. The energy liberated from this oxidation will be used to create adenosine triphosphate from ADP and inorganic phosphate. The matrix is involved with all this, and as well as some of the inner membranes of the mitochondrium. It also uses a proton gradient to affect the use of the production of all of this energy. There are numerous vitamins also involved in the electron transport system. Vitamin A, retinol, is a single transduction activity in the mitochondria. Vitamin B2 acts as FAD, B3 NAD, B9 free radical scavenging. Vitamin C is another antioxidant that protects the mitochondria. CoQ10 is another antioxidant. This is a big part of the cellular energy generating system, and people who take statins and who are genetically predisposed will have a lot of muscle aches and pains that can be improved by taking CoQ10 supplements. Vitamin D is involved in phosphocreatinine recovery. Vitamin E is a membrane antioxidant. It works in the lipid layer. Vitamin K, finally, is also involved in the bacterial electron transport system. We're not certain if it's involved in the human electron transport system or not. So the electron transport system creates ATP from NADH going to NAD, via enzyme-controlled redox reactions between the mitochondrial membranes using proton, chemical, and reduction oxidation gradients, enzymes, cytochrome C, and other factors. Each proton can convert 1 ADP plus inorganic phosphate to ATP. More ATP is generated from the electron transport system than either by glycolysis or the tricarboxylic acid cycle, but the tricarboxylic acid cycle is producing all the NAD and FAD that needs to be oxidized as well. So oxygen plus 2 NADHs gives us 2 ATPs and 2 waters and 2 NADs. Cellular signaling. This again is a rather complex slide that goes into some of the different factors that are involved. Apoptosis is basically cell death. There's gene regulation, gene proliferation. All of these things have to be controlled. And when you've got cancer, you don't have any apoptosis going on. When you don't have proper gene regulation, a lot of other problems can develop. Many chronic diseases come from this way of doing things or not doing them. Major types of cellular signaling involve the endocrine hormone signaling over long distances, paracrine signaling, which happens over much shorter distances, including chemical synaptic activity, juxtacrine signaling, which is adjacent cells with direct contact, such as electrical synapses between nerve cells. Some examples of signals: the endocrine system, thyroid hormones, and sex hormones, for example. The juxtacrine paracrine system involves neurotransmitters, neuropeptides, neuromodulators, and catecholamines. The juxtacrine and paracrine system also are cytokines of the immune system. The classes of signaling molecules include phospholipids, amino acids, lipids, monoamines, proteins, glycoproteins, gases, including hydrogen sulfide and nitrate and carbon monoxide. The receptors must be present on the surface of cell membrane for these receptors and these signaling agents to work. The signal won't work if there's not a receptor on the cell membrane for it. Cyclic adenosine monophosphate, or CAMP, is another example of an intracellular signal activated from a hydrophilic signal onto the cell membrane. So say a thyroid hormone would come along, trigger a receptor on the cell membrane, and that receptor triggers some formation of cyclic AMP, which would then make it into the nucleus of the cell to help regulate genetic activity. Cyclic guanine monophosphate is another one that frequently does the opposite of what CAMP does. Gamma-aminobutyric acid is a neurotransmitter that activates ion channels present on specific cell membranes in the brain to produce action potentials, which depolarize these cells, leading to changes in behavior. Generally, there is an activated membrane receptor that must interact with other proteins inside the cell before the ultimate physiologic cellular effect of the signal occurs. There may be a chain of several such interacting cell proteins after such an interaction. One protein may in turn affect numerous other receptor proteins. This will cause an amplification cascade, where one protein triggers the formation of 10 proteins. Those 10 proteins go on and trigger the formation of 100 others, etc. Hydrophobic signaling molecules, on the other hand, such as the fat-soluble vitamins, can pass directly through the cell membrane and enter the cell nucleus directly. And of course, the sterile hormones, such as the sex hormones, would be part of this class as well. Thank you for being patient with us as we go through the review of the biology. Next, we'll be talking about different vitamins, minerals, supplements, and herbal preparations, and how they work in the body. Thank you.